Thank you. My name is John Kolman. I'm Vice President of Translational Medicine at QPS. Let me begin by saying thank you very, very much for the opportunity to share with you our seat in this crazy ride that we're calling the coronavirus pandemic. As a contract research organization, we support clients around the globe throughout their entire drug development program. Whether that has to do with clinical recruitment in patients in the clinic, whether it has to do with bioanalysis, or even the support that we're providing through our CLIA facilities. So let me just jump into it. I will speak a little bit more quickly so that I can save us some time and get us a bit more on track. Let's just start here. So the COVID-19 global pandemic has been a call to arms for many of the world's biotech and pharmaceutical companies. While we have made some progress, we must continue to pivot and reapply top scientists' creative thinking and material sources to blunt and reverse the scourge which is the coronavirus SARS-CoV-2. This is especially true for QPS and all of us in the vaccine and drug development support industries. QPS quickly recognized that the pharmaceutical industry would require rapid reallocation of resources to SARS-CoV-2 vaccines and therapies. We responded by communicating closely with all of our commercial partners in order to move in lockstep. Our staff were quickly trained in the biology and epidemiology of the virus, as well as the new CDC recommended habits to remain safe and strong. Our facilities were closed to visitors, modified for better airflow processing, and regularly decontaminated with hospital-grade disinfectants. As new information became available, the science was shared and best practices were adjusted. The result, we maintained full-time production using split shifts at 40% occupancy. We have suffered 0 staff infections to date and have been back on our normal daytime operating hours since July 1st. We are an essential business and we have been taxed beyond what was reasonable, but we have answered the call. It was very clear that QPS would play several roles in the fight against the coronavirus. First, in support of sponsored clinical trials using our subject recruitment services and clinics distributed globally, then handing off molecular analysis to our GLP and GCP testing laboratories. Second, we recognized that testing for the SARS-CoV-2 virus and the state of human infection were the first line of defense, and we had all the tools in our high complexity CLIA testing organization to make a difference. QPS has 4 molecular tests for COVID-19 in place or in development. They are the SARS-CoV-2 diagnostic test, a serologic test for SARS-CoV-2 antibodies, a quantitative SARS-CoV-2 genomic test, and an idiopathic upper respiratory infection test. I'm delighted that a number of these tests have already been alluded to by our distinguished speakers prior to me. I will now share more specific information about these 4 tests and how QPS is implementing them to support the fight against SARS-CoV-2. Let's begin with the diagnostic test. The QPS diagnostic test uses the Thermo Fisher Scientific TaqPath COVID-19 multiplex diagnostic solution. It's 2 kits used in succession: nucleic acid extraction followed by RT-qPCR. The test is FDA authorized, Thermo Fisher secured an emergency use authorization, and our service piggybacks on this EUA. The test is multiplexed. There are 3 primer sets directed against 3 targets of SARS-CoV-2, gene ORF1AB, the N protein, and the S protein, as well as a positive control that spiked into the sample prior to extraction. This test recapitulates the original CDC design. It is a qualitative test. At least 2 of 3 positive SARS-CoV-2 targets are required for a positive determination. No amplification of the 3 SARS-CoV-2 primer sets and successful amplification of the spike control is required for a negative determination. The limit of detection, the LOD, in our hands is approximately 50 copies per test. Our preferred collection method is the gold standard, the nasopharyngeal swab, or MP swab. We've also validated the oropharyngeal swab, the OP swab, or the so-called stress test. And as you can see in the cartoon there to the right, the MP test is a very pleasurable experience that everybody should like to look forward to at some point in their lives. Little joke. The testing is performed in our Newark, Delaware facility by highly trained staff. The process is automated for nucleic acid extraction, 384 well plate assembly, and of course the PCR amplification itself. Multiple redundant robotic stations are available for COVID testing. We run the diagnostic test under GCP or CLIA governance. CLIA implementation includes a result reporting 24 hours after receipt of shipment, typically 48 hours after collection, and we are approved to test CLIA samples from all 50 states. Our service includes documentation to all the authorizing medical professionals, the subject when requested, and mandatory reporting to state authorities in which the subject resides. This is a considerable change to our GCP implementation. There's a lot more paperwork, there's a lot more electronic communication, and of course, HIPAA enforcement is quite strict. Diagnostic test scenarios. We test samples in several different scenarios. First, subject or patient samples that are part of a clinical trial and used for inclusion or exclusion. This is done to CLIA, and it's typically contracted by the study sponsor. Second, samples from clinic staff to assure that clinical test conditions do not interfere with study execution. This also is done to CLIA and contracted either by the clinic itself or the study sponsor. And then finally, testing of samples from non-clinical commercial sites in support of workplace testing for staff surveillance and/or contact tracing that is defined and mandated by the commercial leadership. This is also done to CLIA. So-called workplace diagnostic testing is interesting to us as collections are performed in a non-clinical environment. QPS facilitates the process by providing necessary add-on services in addition to the diagnostic test itself. These services include online HIPAA-compliant digital platform for protected health information collection, approved test requisitions and consultation as needed with board-certified physicians for those with positive results. Next, importantly, NP swab kit availability, shipment to the collection site, and finally, on-site sample collection by trained nurses. This end-to-end service is a value add that QPS is proud to offer. We, as a society, need to balance a return to commercial productivity with utmost respect for the safety of those at risk. That starts with everyone that we come in contact with daily, in particular, our staff and our work colleagues. The use of the diagnostic test. This test is for active infection in persons without symptoms or confirmation of the virus's presence in persons with COVID-like symptoms. The frequency of false negative report is about 20%, not the result of shortcomings of the diagnostic test, but rather sample collection. False negative reporting is at a minimum 5 to 8 days after infection, and that is the best point at which to make a collection. The utility of the diagnostic test is twofold. First, identifying infected persons as soon as possible so they can self-quarantine and limit spread of the contagion. And secondly, shortening the quarantine of previously infected or potentially infected persons. The latest CDC guidance lists 10 to 14 days of self-quarantine depending upon exposure type, but 2 diagnostic test negative results separated by 24 hours or more releases the subject from quarantine. The second test is a serologic test for SARS-CoV-2 antibodies. The QPS Serologic test detects antibodies directed against SARS-CoV-2. It is a proprietary test built by our ligand binding assay team, the same team who engineers custom PK and ADA solutions for QPS clients worldwide. The test uses the ELISA platform and is tuned to detect circulating IgG antibodies to the receptor binding domain of the spike protein of SARS-CoV-2. Per current FDA EUA guidance, the test has been evaluated for lack of cross-reactivity to common human coronaviruses 229E, NL63, OC43, HQ1, as well as non-coronaviruses and other ubiquitous antibody reactive or confounding states. The estimated sensitivity of the assay calculated as the percent positive agreement was found to be 92.4%. The estimated specificity of the assay calculated as the NPA, the negative percent agreement, was found to be 98%. Mass-based sensitivity of the positive control was estimated at 285 nanograms per ml. Finally, the test can be used qualitatively or quasi-quantitatively by using a secondary titer evaluation. The qualitative IgG serologic test has been submitted to the FDA for EUA approval. It is currently available to GCP and will be available to CLIA upon EUA approval. An IgM-directed test is under evaluation at this time.
Hey, John, can you finish in 2 minutes?
Yes, absolutely.
All right.
The use of the serologic test is as follows. I'd like to point out just very quickly, the IgG seropositivity reaches a maximum among infected persons about 14 days after infection. It is an indicator of prior SARS-CoV-2 infection. It is very specific. It is not an indicator of current infection. Our state of understanding at this time is that SARS-CoV-2 seropositivity follows infection. It is further understood that seropositivity supports a condition in which the subject is likely to be unlikely to be reinfected and become viremic if re-exposed to the virus. This protected state has been shown to last for 3 months after the initial infection, and it's debated how much longer it might last past that. Taken together, these tests are appropriate for subjects or samples that are part of a clinical trial, as mentioned previously. Secondly, to characterize or better understand the clinical trial population of a clinical trial. And then lastly, it can be used in a non-clinical commercial environment, but it should be taken with a grain of salt. As has been pointed out, it's not 100% sure the value of these protective antibodies at this time. The last two tests, just very quickly. One is a quantitative SARS-CoV-2 genomic test. As like the sero test, this is intended to be proprietary. It is built by the QPS team that engineers all of our custom qPCR assays. And the intent is to support viral load studies as part of clinical trials. Last, the second is an idiopathic upper respiratory infection test. This is also going to be qualitative. It'll be a multiplex qPCR for 20+ viruses and bacteria that are common during the flu season. Targets were chosen specifically for a lack of symptomatic identification during presentation. Flu, SARS-CoV-2, and the common coronaviruses are included. The intent is to separate SARS-CoV-2 patients and anxiety from the regular repertoire of cold and flu agents this fall. Just to conclude, QPS has adjusted and improved its processes to ensure the safety of our staff and the health of our staff, and been able to maintain our laboratories operating at 100% efficiency and capacity. We have developed and are running multiple SARS-CoV-2 test types in support of the global effort to continue to combat the pandemic. Please reach out to us either by contacting your QPS BD, Joy Concepcion, or QPS directly by any of the means listed on the screen. Thank you very much.